Neuroplasticity.
Neuroplasticity, also known as brain plasticity, refers to changes in neural pathways and synapses which are due to changes in behavior, environment and neural processes, as well as changes resulting from bodily injury. Neuroplasticity has replaced a formerly held position that the brain is a physiologically static organ, and explores how, and in which ways, the brain changes throughout life. Neuroplasticity occurs on a variety of levels, ranging from cellular changes due to learning, to large-scale changes involved in cortical remapping in response to injury. The role of neuroplasticity is widely recognized in healthy development, learning, memory, and recovery from brain damage. During most of the 20th century, the consensus among neuroscientists was that brain structure is relatively immutable after a critical period during early childhood. This belief has been challenged by findings revealing that many aspects of the brain remain plastic even into adulthood. Hubel and Wiesel had demonstrated that ocular dominance columns in the lowest neocortical visual area, V1, were largely immutable after the critical period in development. Critical periods also were studied with respect to language. The resulting data suggested that sensory pathways were fixed after the critical period. However, studies determined that environmental changes could alter behavior and cognition by modifying connections between existing neurons and via neurogenesis in the hippocampus and other parts of the brain, including the cerebellum. Decades of research have now shown that substantial changes occur in the lowest neocortical processing areas, and that these changes can profoundly alter the pattern of neuronal activation in response to experience. Neuroscientific research indicates that experience can actually change both the brain's physical structure, anatomy, and functional organization, physiology. Neuroscientists are currently engaged in a reconciliation of critical period studies demonstrating the immutability of the brain after development with the more recent research showing how the brain can, and does, change.
Neurobiology.
One of the fundamental principles of how neuroplasticity functions is linked to the concept of synaptic pruning, the idea that individual connections within the brain are constantly being removed or recreated, largely dependent upon how they are used. This concept is captured in the aphorism, "Neurons that fire together, wire together / neurons that fire apart, wire apart." If there are two nearby neurons that often produce an impulse simultaneously, their cortical maps may become one. This idea also works in the opposite way, that is, that neurons which do not regularly produce simultaneous impulses will form different maps.
Cortical maps.
Cortical organization, especially for the sensory systems, is often described in terms of maps. For example, sensory information from the foot projects to one cortical site and the projections from the hand target in another site. As the result of the somatotopic organization of sensory inputs to the cortex, cortical representation of the body resembles a map or homunculus. In the late 1970s and early 1980s, several groups began exploring the impacts of removing portions of the sensory inputs. Michael Merzenich, John Cause and Doug Rasmussen used the cortical map as their dependent variable. They found, and this has been since corroborated by a wide range of labs, that if the cortical map is deprived of its input it will become activated at a later time in response to other, usually adjacent inputs. Merzenich's 1984 study involved the mapping of owl monkey hands before and after amputation of the third digit. Before amputation, there were 5 distinct areas, one corresponding to each digit of the experimental hand. 62 days following amputation of the third digit, the area in the cortical map formerly occupied by that digit had been invaded by the previously adjacent second and fourth digit zones. The areas representing digit 1 and 5 are not located directly beside the area representing digit 3, so these regions remained, for the most part, unchanged following amputation. This study demonstrates that only those regions bordering a certain area will invade it to alter the cortical map. In the somatic sensory system, in which this phenomenon has been most thoroughly investigated, J.T. Wall and J. Zhu have traced the mechanisms underlying this plasticity. Reorganization is not cortically emergent, but occurs at every level in the processing hierarchy. This produces the map changes observed in the cerebral cortex. Merzenich and William Jenkins 1990, initiated studies relating sensory experience, without pathological perturbation, to cortically observed plasticity in the primate somatosensory system, with the finding that sensory sites activated in an attended operant behavior increase in their cortical representation. Shortly thereafter, Ford Ebner and colleagues 1994, made similar efforts in the rodent whisker-barrel cortex also somatic sensory system. These two groups largely diverged over the years. The rodent whisker-barrel efforts became a focus for Ebner, Matthew Diamond, Michael Armstrong James, Robert Sachdove, Kevin Fox, and great inroads were made in identifying the locus of change as being at cortical synapses expressing NMDA receptors, and in implicating cholinergic inputs as necessary for normal expression. However, the rodent studies were poorly focused on the behavioral end, and Ron Frostig and Daniel Polly 1999, 2004, identified behavioral manipulations as causing a substantial impact on the cortical plasticity in that system. Merzenich and D.T. Blake 2002, 2005, 2006, went on to use cortical implants to study the evolution of plasticity in both the somatosensory and auditory systems. Both systems show similar changes with respect to behavior. When a stimulus is cognitively associated with reinforcement, its cortical representation is strengthened and enlarged. In some cases, cortical representations can increase 2 to 3-fold in 1-2 days at the time at which a new sensory motor behavior is first acquired, and changes are largely finished within at most a few weeks. Control studies show that these changes are not caused by sensory experience alone, they require learning about the sensory experience, and are strongest for the stimuli that are associated with reward, and occur with equal ease in operant and classical conditioning behaviors. An interesting phenomenon involving cortical maps is the incidence of phantom limbs. Phantom limbs are experienced by people that have undergone amputations in hands, arms, and legs, but it is not limited to extremities. Although the neurological basis of phantom limbs is still not entirely understood, it is believed that cortical reorganization plays an important role. Norman Doidge, following the lead of Michael Merzenich, separates manifestations of neuroplasticity into adaptations that have positive or negative behavioral consequences. For example, if an organism can recover after a stroke to normal levels of performance, that adaptiveness could be considered an example of positive plasticity. Changes such as an excessive level of neuronal growth leading to spasticity or tonic paralysis, or an excessive release of neurotransmitters in response to injury which could kill nerve cells, would have to be considered negative plasticity. In addition, drug addiction and obsessive-compulsive disorder are deemed examples of negative plasticity by Dr. Doidge, as the synaptic rewiring resulting in these behaviors is also highly maladaptive. A 2005 study found that the effects of neuroplasticity occur even more rapidly than previously expected. Medical students' brains were imaged during the period when they were studying for their exams. In a matter of months, the students' gray matter increased significantly in the posterior and lateral parietal cortex.
Applications and example. Treatment of brain damage.
A surprising consequence of neuroplasticity is that the brain activity associated with a given function can move to a different location. This can result from normal experience and also occurs in the process of recovery from brain injury. Neuroplasticity is the fundamental issue that supports the scientific basis for treatment of acquired brain injury with goal-directed experiential therapeutic programs in the context of rehabilitation approaches to the functional consequences of the injury. The adult brain is not entirely hardwired with fixed neuronal circuits. There are many instances of cortical and subcortical rewiring of neuronal circuits in response to training as well as in response to injury. There is solid evidence that neurogenesis birth of brain cells occurs in the adult, mammalian brain, and such changes can persist well into old age. The evidence for neurogenesis is mainly restricted to the hippocampus and olfactory bulb, but current research has revealed that other parts of the brain, including the cerebellum, may be involved as well. In the rest of the brain, neurons can die, but they cannot be created. However, there is now ample evidence for the active, experience-dependent reorganization of the synaptic networks of the brain involving multiple interrelated structures including the cerebral cortex. The specific details of how this process occurs at the molecular and ultrastructural levels are topics of active neuroscience research. The manner in which experience can influence the synaptic organization of the brain is also the basis for a number of theories of brain function, including the general theory of mind and epistemology referred to as neural Darwinism and developed by immunologist Nobel laureate Gerald Edelman. The concept of neuroplasticity is also central to theories of memory and learning that are associated with experience-driven alteration of synaptic structure and function in studies of classical conditioning in invertebrate animal models such as aplysia. This latter program of neuroscience research has emanated from the groundbreaking work of another Nobel laureate, Eric Kendall, and his colleagues at Columbia University College of Physicians and Surgeons. Paul Bach-Wierita, deceased in 2006, was the father of sensory substitution and brain plasticity. In working with a patient whose vestibular system had been damaged, he developed BrainPort, a machine that replaces her vestibular apparatus and sent balance signals to her brain from her tongue. After she had used this machine for some time, it was no longer necessary, as she regained the ability to function normally. Her balancing act days were over. Plasticity is the major explanation for the phenomena. Because her vestibular system was disorganized and sending random rather than coherent signals, the apparatus found new pathways around the damaged or blocked neural pathways, helping to reinforce the signals that were sent by remaining healthy tissues. Bach-Wierita explained plasticity by saying, "If you are driving from here to Milwaukee and the main bridge goes out, first you are paralyzed. Then you take old secondary roads through the farmland. Then you use these roads more. You find shorter paths to use to get where you want to go, and you start to get there faster. These secondary neural pathways are unmasked or exposed and strengthened as they are used." The unmasking process is generally thought to be one of the principal ways in which the plastic brain reorganizes itself. Rambuto's group found that if a small stroke and infarction is induced by obstruction of blood flow to a portion of a monkey's motor cortex, the part of the body that responds by movement will move when areas adjacent to the damaged brain area are stimulated. In one study, intracortical microstimulation (ICMS) mapping techniques were used in 9 normal monkeys. Some underwent ischemic infarction procedures and the others, ICMS procedures. The monkeys with ischemic infarctions retained more finger flexion during food retrieval, and after several months this deficit returned to preoperative levels. With respect to the distal forelimb representation, post-infarction mapping procedures revealed that movement representations underwent reorganization throughout the adjacent, undamaged cortex. Understanding of interaction between the damaged and undamaged areas provides a basis for better treatment plans in stroke patients. Current research includes the tracking of changes that occur in the motor areas of the cerebral cortex as a result of a stroke. Thus, events that occur in the reorganization process of the brain can be ascertained. Duto is also involved in studying the treatment plans that may enhance recovery from strokes, such as physiotherapy, pharmacotherapy and electrical stimulation therapy. Neuroplasticity is gaining popularity as a theory that, at least in part, explains improvements in functional outcomes with physical therapy post-stroke. Rehabilitation techniques that have evidence to suggest cortical reorganization as the mechanism of change include constraint-induced movement therapy, functional electrical stimulation, treadmill training with body weight support, and virtual reality therapy. Robot-assisted therapy is an emerging technique, which is also hypothesized to work by way of neuroplasticity, though there is currently insufficient evidence to determine the exact mechanisms of change when using this method. John Coz, a professor at Vanderbilt University, has been able to show how somatosensory area 3B and ventroposterior VP nucleus of the thalamus are affected by long-standing unilateral dorsal column lesions at cervical levels in macaque monkeys. Adult brains have the ability to change as a result of injury, but the extent of the reorganization depends on the extent of the injury. His recent research focuses on the somatosensory system, which involves a sense of the body and its movements using many senses. Usually when people damage the somatosensory cortex, impairment of the body perceptions are experienced. He is trying to see how these systems, somatosensory, cognitive, motor systems, are plastic as a result of injury. One of the most recent applications of neuroplasticity involves work done by a team of doctors and researchers at Emory University, specifically Dr. Donald Stein, who has been in the field for over 3 decades, and Dr. David Wright. This is the first treatment in 40 years that has significant results in treating traumatic brain injuries while also incurring no known side effects and being cheap to administer. Dr. Stein noticed that female mice seemed to recover from brain injuries better than male mice. Also in females, he noticed that at certain points in the estrous cycle females recovered even more. After lots of research, they attributed this difference due to the levels of progesterone. The highest level of progesterone present led to the fastest recovery of brain injury in these mice. They developed a treatment that includes increased levels of progesterone injections to give to brain-injured patients. And administration of progesterone after traumatic brain injury, TBI, and stroke reduces edema, inflammation, and neuronal cell death, and enhance spatial reference memory and sensory motor recovery. In their clinical trials, they had a group of severely injured patients that after the 3 days of progesterone injections had a 60% reduction in mortality. Sam was in a horrific car accident that left him with marginal brain activity. According to the doctors, he was 1 point away from being brain dead. His parents decided to have him participate in Dr. Stein's clinical trial, and he was given the 3-day progesterone treatment. 3 years after the accident, he had achieved an inspiring recovery with no brain complications and the ability to live a healthy, normal life. Stein has done some studies in which beneficial effects have been seen to be similar in aged rats to those seen in youthful rats. As there are physiological differences in the 2 age groups, the model was tweaked for the elderly animals by reducing their stress levels with increased physical contact. During the surgery, anesthesia was kept at a higher oxygen level with lower overall isoflurane percentage, and the aged animals were given subcutaneous lactated Ringer's solution post-surgery to replace fluids lost through increased bleeding. The promising results of progesterone treatments could have a significant impact on the clinical management of TBI. These treatments have been shown to work on human patients who receive treatment soon after the TBI. However, Dr. Stein now focuses his research on those persons who have long-standing traumatic brain injury in order to determine if progesterone treatments will assist them in the recovery of lost functions as well.
Treatment of learning difficulties.
Michael Merzenich developed a series of plasticity-based computer programs known as Fast Forward. Fast Forward offers 7 brain exercises to help with the language and learning deficits of dyslexia. In a recent study, experimental training was done in adults to see if it would help to counteract the negative plasticity that results from age-related cognitive decline (ARCD). The ET design included 6 exercises designed to reverse the dysfunctions caused by ARCD in cognition, memory, motor control, and so on. After use of the ET program for 8-10 weeks, there was a significant increase in task-specific performance. The data collected from the study indicated that a neuroplasticity-based program could notably improve cognitive function and memory in adults with ARCD.
Neuroplasticity during operation of brain-machine interfaces.
Brain-machine interface (BMI) is a rapidly developing field of neuroscience. According to the results obtained by Mikhail Lebdev, Miguel Nicolelis and their colleagues, operation of BMIs results in incorporation of artificial actuators into brain representations. The scientists showed that modifications in neuronal representation of the monkey's hand and the actuator that was controlled by the monkey brain occurred in multiple cortical areas while the monkey operated a BMI. In these single-day experiments, monkeys initially moved the actuator by pushing a joystick. After mapping out the motor neuron ensembles, control of the actuator was switched to the model of the ensemble so that the brain activity, and not the hand, directly controlled the actuator. The activity of individual neurons and neuronal populations became less representative of the animal's hand movements while representing the movements of the actuator. Presumably as a result of this adaptation, the animals could eventually stop moving their hands yet continue to operate the actuator. Thus, during BMI control, cortical ensembles plastically adapt, within tens of minutes, to represent behaviorally significant motor parameters, even if these are not associated with movements of the animal's own limb. Active laboratory groups include those of John Donahue at Brown, Richard Anderson at Caltech, Krishna Shenoy at Stanford, Nicholas Hatsopoulos of University of Chicago, Andy Schwartz at University of Pittsburgh, Sandro Masa-Evaldi at Northwestern and Miguel Nicolelis at Duke. Donahue and Nicolelis groups have independently shown that animals can control external interfaces in tasks requiring feedback, with models based on activity of cortical neurons, and that animals can adaptively change their minds to make the models work better. Donahue's group took the implants from Richard Norman's lab at the Utah Array, and improved it by changing the insulation from polyamide to parylene C, and commercialized it through the company Cyberkinetics. These efforts are the leading candidate for the first human trials on a broad scale for motor cortical implants to help quadriplegic or locked-in patients communicate with the outside world.
Sensory prostheses.
Neuroplasticity is involved in the development of sensory function. The brain is born immature and it adapts to sensory inputs after birth. In the auditory system, congenital hearing impairment, a rather frequent inborn condition affecting 1 of 1,000 newborns, has been shown to affect auditory development, and implantation of a sensory prosthesis activating the auditory system has prevented the deficits and induced functional maturation of the auditory system due to a sensitive period for plasticity. There is also a sensitive period for such intervention within the first 2-4 years of life. Consequently, in prelingually deaf children, early cochlear implantation as a rule allows to learn mother language and acquire acoustic communication.
Phantom limbs.
The experience of phantom limbs is a phenomenon in which a person continues to feel pain or sensation within a part of their body which has been amputated. This is strangely common, occurring in 60-80% of amputees. An explanation for this refers to the concept of neuroplasticity, as the cortical maps of the removed limbs are believed to have become engaged with the area around them in the postcentral gyrus. This results in activity within the surrounding area of the cortex being misinterpreted by the area of the cortex formerly responsible for the amputated limb. The relationship between phantom limbs and neuroplasticity is a complex one. In the early 1990s versus Ramachandran theorized that phantom limbs were the result of cortical remapping. However, in 1995 Herdefloor and her colleagues demonstrated that cortical remapping occurs only in patients who have phantom pain. Her research showed that phantom limb pain rather than referred sensations was the perceptual correlate of cortical reorganization. This phenomenon is sometimes referred to as maladaptive plasticity. In 2009 Lorimer Mosley and Peter Brugger carried out a remarkable experiment in which they encouraged arm amputee subjects to use visual imagery to contort their phantom limbs into impossible configurations. 4 of the 7 subjects succeeded in performing impossible movements of the phantom limb. This experiment suggests that the subjects had modified the neural representation of their phantom limbs and generated the motor commands needed to execute impossible movements in the absence of feedback from the body. The authors stated that, in fact, this finding extends our understanding of the brain's plasticity because it is evidence that profound changes in the mental representation of the body can be induced purely by internal brain mechanisms. The brain truly does change itself.
Chronic pain.
Individuals who suffered from chronic pain experience prolonged pain at sites that may have been previously injured, yet are otherwise currently healthy. This phenomenon is related to neuroplasticity due to a maladaptive reorganization of nervous system, both peripherally and centrally. During the period of tissue damage, noxious stimuli and inflammation cause an elevation of nociceptive input from the periphery to the central nervous system. Prolonged nociception from periphery will then elicit a neuroplastic response at the cortical level to change its somatotopic organization for the painful site, inducing central sensitization. For instance, Individuals experiencing complex regional pain syndrome demonstrate a diminished cortical somatotopic representation of the hand contralaterally as well as a decreased spacing between the hand and the mouth. Additionally, chronic pain has been reported to significantly reduce the volume of gray matter in the brain globally, and more specifically at the prefrontal cortex and right thalamus. However, following treatment, these abnormalities in cortical reorganization and gray matter volume are resolved, as well as their symptoms. Similar results have been reported for phantom limb pain, chronic low back pain and carpal tunnel syndrome.
Meditation.
A number of studies have linked meditation practice to differences in cortical thickness or density of gray matter. One of the most well-known studies to demonstrate this was led by Sarah Laser, from Harvard University, in 2000. Richard Davidson, a neuroscientist at the University of Wisconsin, has led experiments in cooperation with the Dalai Lama on effects of meditation on the brain. His results suggest that long-term, or short-term practice of meditation results in different levels of activity in brain regions associated with such qualities as attention, anxiety, depression, fear, anger, the ability of the body to heal itself, and so on. These functional changes may be caused by changes in the physical structure of the brain.
Fitness and exercise.
In a 2009 study, scientists made two groups of mice swim a water maze, and then in a separate trial subjected them to an unpleasant stimulus to see how quickly they would learn to move away from it. Then, over the next 4 weeks they allowed one group of mice to run inside their rodent wheels, an activity most mice enjoy, while they forced the other group to work harder on mini treadmills at a speed and duration controlled by the scientists. They then tested both groups again to track their learning skills and memory. Both groups of mice improved their performances in the water maze from the earlier trial. But only the extra work treadmill runners were better in the avoidance task, a skill that, according to neuroscientists, demands a more complicated cognitive response. The mice who were forced to run on the treadmills showed evidence of molecular changes in several portions of their brains when viewed under a microscope, while the voluntary wheel runners had changes in only one area. Our results support the notion that different forms of exercise induce neuroplasticity changes in different brain regions, Xiang Zhen, a professor of physiology and an author of the study, said.
Human echolocation.
Human echolocation is a learned ability for humans to sense their environment from echoes. This ability is used by some blind people to navigate their environment and sense their surroundings in detail. Studies in 2010 and 2011 using functional magnetic resonance imaging techniques have shown that parts of the brain associated with visual processing are adapted for the new skill of echolocation. Studies with blind patients, for example, suggest that the click echoes heard by these patients were processed by brain regions devoted to vision rather than audition.
Etymology.
Plasticity was first applied to behavior in 1890 by William James in The Principles of Psychology, though the idea was largely neglected for the next 50 years. The first person to use the term neuroplasticity appears to have been the Polish neuroscientist Jerzy Konarski.
History. Proposal.
Until around the 1970s, an accepted idea across neuroscience was that the nervous system was essentially fixed throughout adulthood, both in terms of brain functions, as well as the idea that it was impossible for new neurons to develop after birth. In 1793, Italian anatomist Michele Vincenzo Malacarne described experiments in which he paired animals, trained one of the pair extensively for years, and then dissected both. He discovered that the cerebellums of the trained animals were substantially larger. But, these findings were eventually forgotten. The idea that the brain and its functions are not fixed throughout adulthood was proposed in 1890 by William James in The Principles of Psychology, though the idea was largely neglected.
Research and discovery.
In 1923, Carl Ashley conducted experiments on rhesus monkeys which demonstrated changes in neuronal pathways, which he concluded to be evidence of plasticity, although despite this, as well as further examples of research suggesting this, the idea of neuroplasticity was not widely accepted by neuroscientists. However, more significant evidence began to be produced in the 1960s and after, notably from scientists including Paul Bach-Wierita, Michael Merzenich along with John Coase, as well as several others. In the 1960s, Paul Bach-Wierita invented a device that allowed blind people to read, perceive shadows, and distinguish between close and distant objects. This machine was one of the first and boldest applications of neuroplasticity. The patient sat in an electrically stimulated chair that had a large camera behind it which scanned the area, sending electrical signals of the image to 400 vibrating stimulators on the chair against the patient's skin. The 6 subjects of the experiment were eventually able to recognize a picture of the supermodel Twiggy. It must be emphasized that these people were congenitally blind and had previously not been able to see. Bach-Wierita believed in sensory substitution. If one sense is damaged, your other senses can sometimes take over. He thought skin and its touch receptors could act as a retina using one sense for another. In order for the brain to interpret tactile information and convert it into visual information, it has to learn something new and adapt to the new signals. The brain's capacity to adapt implied that it possessed plasticity. He thought, we see with our brains, not with our eyes. A tragic stroke that left his father paralyzed inspired Bach-Wierita to study brain rehabilitation. His brother, a physician, worked tirelessly to develop therapeutic measures which were so successful that the father recovered complete functionality by age 68 and was able to live a normal, active life which even included mountain climbing. His father's story was first-hand evidence that a late recovery could occur even with a massive lesion in an elderly person. He found more evidence of this possible brain reorganization with Shepherd Ivory Franz's work. One study involved stroke patients who were able to recover through the use of brain-stimulating exercises after having been paralyzed for years. Franz understood the importance of interesting, motivating rehabilitation. Under conditions of interest, such as that of competition, the resulting movement may be much more efficiently carried out than in the dull, routine training in the laboratory. Franz, 1921, pg 93. This notion has led to motivational rehabilitation programs that are used today. Michael Merzenich is a neuroscientist who has been one of the pioneers of neuroplasticity for over 3 decades. He has made some of the most ambitious claims for the field, that brain exercises may be as useful as drugs to treat diseases as severe as schizophrenia, that plasticity exists from cradle to the grave, and that radical improvements in cognitive functioning, how we learn, think, perceive, and remember are possible even in the elderly. Merzenich's work was affected by a crucial discovery made by David Hubel and Torsten Wiesel in their work with kittens. The experiment involved sewing one eye shut and recording the cortical brain maps. Hubel and Wiesel saw that the portion of the kitten's brain associated with the shut eye was not idle, as expected. Instead, it processed visual information from the open eye. It was as though the brain didn't want to waste any cortical real estate and had found a way to rewire itself. This implied neuroplasticity during the critical period. However, Merzenich argued that neuroplasticity could occur beyond the critical period. His first encounter with adult plasticity came when he was engaged in a postdoctoral study with Clinton Woosley. The experiment was based on observation of what occurred in the brain when one peripheral nerve was cut and subsequently regenerated. The two scientists micromapped the hand maps of monkey brains before and after cutting a peripheral nerve and sewing the ends together. Afterwards, the hand map in the brain that was expected to be jumbled was nearly normal. This was a substantial breakthrough. Merzenich asserted that if the brain map could normalize its structure in response to abnormal input, the prevailing view that we are born with a hardwired system had to be wrong. The brain had to be